Hello, and welcome to a very special episode of the podcast Byzantium and Friends. This is going to be an unusual one with a lot more friends this time. And I will be hosting this discussion, but I didn't organize it. This was an idea pitched to me by longstanding friend of the podcast, Tina Sessa, professor of history at The Ohio State University, who is joining me here today to tell us about the background and how this came to be and what we're talking about, so that you can all follow the conversation more easily. Tina, why don't you tell us where this is coming from?
Thanks, Anthony. Hello, everyone. I am so excited to present to you today the first of two podcasts that were organized by myself, whom, as many of you know, is a scholar of cultural history, and my colleague, Tim Newfield, who teaches at Georgetown University. Tim is a historical epidemiologist and an environmental historian. Tim and I decided we wanted to put together a project that would help historians in particular learn more and become more familiar with paleoscience, with the study of the natural sciences as applied to the ancient world. There are two podcasts. The first one you are about to hear is on the topic of human paleogenetics, human aDNA, and the study of migration in late antiquity. It's a roundtable that brings together a number of different kinds of scholars, a computational geneticist, an anthropologist, as well as several historians who work closely on this field. It also involves several archaeologists. I hope you enjoy it, and please stay tuned for the next iteration, which will involve pathogen aDNA and the study of disease in late antiquity. Thanks.
Thank you, Tina. And I should say that we introduced the participants in this discussion at the beginning of the discussion, so we won't do so now. And everyone, look forward to the next installment in this series of two, which will be on the pathogen DNA. And that was a fun discussion too. All right, let's go straight to the roundtable then. Hello, and welcome to our discussion of paleogenetics. I am Anthony Kaldellis. I am a professor of classics at the University of Chicago. My area of specialty is Byzantium, also known as the Eastern Roman Empire. And I'm going to ask our respondents today to introduce themselves so that you in the audience know who they are. I will start with Hannah. Can you please introduce yourself?
Yeah, absolutely. And thanks so much for having me. I'm a postdoc at the University of Chicago. I work jointly between the Department of Human Genetics and ISAC, which is the Institute for the Study of Ancient Cultures on campus. And my research focuses on using genetic and archaeological information to study mobility in the Mediterranean in the Iron Age, the Roman period, and late antiquity.
Thank you, Hannah. Krishna?
Hi, I'm Krishna Veeramah. I'm an associate professor at Stony Brook University in the Department of Ecology and Evolution. I'm an evolutionary genomicist.
Thank you, Krishna. Patrick?
Thanks for this opportunity. I'm Patrick Geary. I'm emeritus professor of medieval history at the Institute for Advanced Study in Princeton, and I am co-PI with a geneticist, a historian, an archaeologist, of a large European Research Council project that is combining genomic, archaeological, isotopic, historical data to understand the Carpathian Basin at the end of Antiquity and the early Middle Ages.
Thank you, Patrick. Janet?
I'm Janet Kay. I'm a lecturer in the Department of Art and Archaeology at Princeton University, and I work primarily on late Roman and early medieval Britain.
Thank you, Janet. István?
Hello, I'm István Koncz. I'm an archaeologist from the Eötvös Loránd University, Budapest, Hungary. My area of expertise is early medieval archaeology in Europe, like mostly focused on the Carpathian Basin between the 4th and 7th, 8th centuries.
Thank you, István. And last but not least, Brian.
Hey, I'm Brian Swain. I'm associate professor of history at Kennesaw State University. I study late Roman history and the barbarians.
Thank you, Brian. So let's just jump right into it, and I will ask the first question. So the first ancient human genome was sequenced and published in 2010. From this single genome, the field of paleogenomics has expanded rapidly. So we are now in the midst of a scientific revolution with over 10,000 ancient genomes published. What stand out to you as the major technological and methodological advances in the field? Where are we and how did we get here? What major obstacles remain? Hannah, do you want to start us off here?
Yeah, sure. Thanks so much. And yeah, the 2010 was a really big year for ancient DNA, both because of, as you mentioned, the first ancient modern human genome was published by Rasmussen et al. And then the first Neanderthal, so archaic human ancient genome, there were 2 papers on that by Green et al. And Noonan et al. And while today we might sort of think it's common knowledge that modern humans carry Neanderthal genes, these were the first papers that showed that. So, even these relatively recent papers have had these big impacts on the field. And a few things that have sort of changed sort of in terms of the research since that time are a lot of progress in terms of targeting which tissues to aim for when you're doing wet lab work. So I do a lot of work in the wet lab where you're sort of wearing the full Tyvek suit and the face mask and multiple pairs of gloves. And essentially that's not to protect you from anything you're handling, but it's to protect the sample from your DNA. So you're trying not to contaminate anything that you're working with. So there's been developments in preventing contamination so that you can go into the lab and do this work with confidence. And also what tissue to target. Initially, it was thought that long bones might be the best tissues to target for extracting ancient DNA, but it turns out that teeth and the petrous bone, which is an inner ear bone in the crania, are actually the best places to target. And what's good about this is it sort of improves the success rate of material that you're working with and also minimizes destruction because you have these higher success rates. So you're able to take less and feel more confident about the chance of success, which is of course still low because we're working with ancient and degraded material. But these have been ways that it's been optimized. There's even been research into minimally invasive methods such as sort of soaking teeth in buffer so that there isn't structural damage. You would dissolve a little bit of the DNA, but then there's not structural or morphological damage to the tooth itself. A few other things is there's been many different lab work developments. So one that I'm particularly fond of is a treatment called UDG. And essentially what that does is ancient DNA or DNA gets damaged over time. And these damages accumulate and then are sequenced. And they actually look like a different base pair than what is actually there in the genome. So when you sequence them, it can give you a false read in your sequence data. And so this is a treatment that essentially cleaves the DNA at any of these damage sites, and it lets you get a higher quality genome or a genome with fewer calls that are, that are sort of affected by this damage. So that's been a really cool development. And then a few things that I've been really excited about is that there's a lot of labs that are sort of entering the field with different perspectives, different goals. And so sort of, a lot of the choices that were made early on in the development of the field have sort of structured and become entangled with how we all do ancient DNA. And so it's been really exciting to see labs being creative and challenging these, both in the lab work side and the computational side. So I'm really excited for sort of the coming years to see how people are handling these challenges and thinking about new ways to solve them. And maybe this is a good time to hand off to some computational people to answer what's happening on that side of the work.
Thank you, Hannah. Krishna?
Yeah, I mean, probably, the first ancient DNA was extracted by Svante Pääbo in 85. The likelihood was that was probably contamination, in truth. But it was a landmark kind of thing, the first human ancient DNA at least. For a long while, ancient DNA as a field was very speculative. There were a lot of claims in the 90s, in particular, of getting DNA from all sorts of organisms, dinosaurs, for example, which we now think is pretty impossible. And so I won't say it was a joke, but it was very, very speculative. And there were just a handful of labs that really people thought could do this effectively. You needed these very, very clean laboratory conditions. The thing that changed all of that was next generation sequencing. That completely revolutionized, it's revolutionized all of genomics, but in particular, it's revolutionized ancient DNA. Because before that, we were reliant on a procedure called PCR. Probably everyone's heard of PCR now from COVID and things like that. But it's a fundamental molecular biology technique where we can target specific parts of DNA that we're interested in and amplify the DNA over and over again so that we can use it to do all sorts of cool things, but it wasn't very suited for ancient DNA because it required DNA that was of a certain length of size. And ancient DNA tends to be very fragmented. Like you die, and then suddenly the enzymes that are usually there to maintain all your DNA in your cells disappear, and your DNA starts to degrade, and they get smaller and fragmented. And PCR was very, very poor at kind of picking up real ancient DNA, and it would tend to pick up modern contaminated DNA. So most kind of DNA that you'd get from these old ancient DNA studies was contamination, modern contamination. Who was in the lab, who handled the chemicals, who handled the bones. Next generation sequencing has completely changed everything. Like all the stuff with Petrus bone and UDG, they're all pretty cool, but none of this works without next generation sequencing. And that was the ability to just sequence short fragments in huge, huge numbers, billions and billions of fragments in one time. And yeah, without a doubt, that's the thing that kind of allowed sequencing of the Neanderthal genome and so on and so forth. So that's the most important technological thing, I would say. From terms of methodology, being able to manage big data has been a big part of what people that work with ancient DNA had to kind of wrangle with, because now we're dealing with huge amounts of sequencing data. And so having a lot of computational background to do that is actually part of the necessary skill set now. You need to basically understand how computers work and how servers work and things like that. And we still haven't actually got all around that. We're not exploiting all the information theoretically that's in genetic data. But, you know, with machine learning and kind of these kind of things, we are getting a handle on it.
Okay, so unless anyone wants to jump in and add something to that, I will go to our second question. So what is population genetics, and how does it relate to the study of ancient DNA and ancient populations? Does paleogenetics differ from modern genetics? And if so, how? I'll just mention here anecdotally. So one problem that I've come across in studies of this sort is that they sometimes tend to identify a certain modern population as being indicative as a kind of baseline for, quote, Greek DNA or Slavic DNA, and then they compare it to other parts of the population in those countries or what they find in medieval graves. And so I was just wondering, what are the methodological challenges that are posed by those kinds of approaches? So Krishna, why don't you start us off with that?
So yeah, so population genetics, you know, at its heart is trying to understand why populations, within populations, between populations have the patterns of genetic variation that's there. That's fundamentally what we're trying to get at. It was the critical field that bridged the gap of Darwin to genetics. So it's basically why we had the modern synthesis of natural selection. So it's a field that emerged from Wright and Fisher and Haldane and these big people from the 50s and 60s. Absolutely fundamental for us, kind of evolution being accepted in academia and in general. When people are interested in population genetics, they're interested in more, what are the kind of things, how has natural selection shaped our genomes? That's what most people who are interested in population genetics are interested in. You know, how has this particular mutation become advantageous and managed to find its way in every single person, like lactase persistence, for example, in certain populations and these kind of things. But underlying that, there is this knowledge that you have to know not just the kind of how the genome is shaped by natural selection, but also processes like population history, that there's kind of the demographic history shapes the genetic variation we see within and between individuals. And from kind of ancient DNA perspective, people that are interested in history have kind of used the principles of population genetics to understand historical facets of modern populations. What demographic processes have resulted in the modern variation we see today? What kind of population divergences, splits, migrations in somewhat simplistic models from a historical perspective, but from a population genetics perspective, they capture most of the information we need. So that's what population genetics is and how it's been used from a kind of from an ancient DNA perspective. It wasn't designed to address ancient DNA. It was designed to just understand kind of selective forces, really. So if I break down the rest of the questions, does paleogenetics differ from modern genetics? I mean, fundamentally, no. When we're thinking about and it somewhat depends on the question you're asking. So we can think about paleogenetics from the perspective of, are we asking about the populations that we're looking at in the past? So for example, are we trying to understand the genetic variation from 5th century Germans, for example? We get DNA from those individuals. Then the principles of population genetics are exactly the same as if we were to do this on modern populations. The only issue that you have is the data is a little messier. The other way that you can use ancient DNA and paleogenetics, which is the study of ancient genomes, is we can better understand modern genetic variation by anchoring it with the genetic variation of past populations. So there's these two different perspectives of why you might want to use ancient genomes or paleogenetics.
Thank you. Patrick?
Well, from the perspective of a historian, my sense is that while there is no fundamental difference, there is simply population genetics applied in different ways. When we're talking about historical period, and in particular the period that your podcast focuses on, which is late antiquity, we're talking about populations that are enormously similar. We're not really looking at evolution in any significant way. We're trying to tease out very, very subtle differences within a genetic pea soup that is very similar across certainly Western Central Europe and to a great extent beyond. So part of the issue is, as Krishna says, the kind of questions we're asking, we're sort of hitching ourselves onto a discipline that has other goals in order to try to tease out these very, very minor differences. Now, you asked about this question of Greek DNA or Slavic DNA, and we'll talk about this, I'm sure, and I think that István will have a lot to say about this. This is simply poor practice. This is a major issue that we encounter constantly, that people are taking ethnic labels and attaching them to genetic populations. That is simply mixing categories that have for which there is no justification. What we're doing technically when we are creating a principal component analysis plot, that is, we are taking our ancient individuals and trying to disaggregate them in a two-dimensional on a two-dimensional axis to see where they cluster and where they separate. Until recently, and one has had to use, for technical reasons that Krishna can go into if necessary, but had to use modern populations. But those modern populations are not ethnic populations. They are geographical populations. So to say it's Greek DNA, no, it's not Greek DNA. It's if this person were living today, there is a good likelihood that they would have lived in that region, which today is Greece. Of course, 1,000 years ago, it wasn't Greece. And parts of it were Greece, and then they weren't Greek, and then they were Turkey, and they were Ottoman. So this is not an ethnic connection. Same thing with Slavic. This is just poor use. Unfortunately, it is very common. And there are a lot of heritage studies that we need because of this growing database. We have to use these population groups, and we have to try to go back and figure out why did they label that? What was behind that mislabeling, and try to come up with a geographical answer. Increasingly, though, at least in our work, we're trying to get away from this the use of modern populations. You mentioned that there are now some 10,000 ancient genomes. Our project alone is now doing over 6,500 genomes. So we're really getting a very, very large, dense, and thick genomic profile of certain areas. It's absolutely terrible in places like France. We have no data whatsoever. In Central Asia, Eastern Asia, it exists, but it's difficult to obtain. But otherwise, we're doing pretty well. So we're going to be able to get away from that simplistic modern projection. And we're already doing that with considerable success. So the problem that you mentioned is just poor methodology. There's nothing intrinsic to population genetics or the methods that we're using. It's just people who haven't reflected sufficiently on what is an ethnic label that creates the problem. And I'm sure we'll hear a lot more about that from people.
Thank you, Patrick. István?
If I may add just a small comment to what Patrick has said about labeling data sets. So what's basically happening is that, let's say you have a first, second century data set from the Caucasus, and it is labeled as, let's say, Alans, without proper archaeological context, sometimes even proper dating. And what we are trying to do in our project, but I think generally in the field, is that moving away from these labels and really like tying it to a geographical location and basically a date, like a general date, if you have something like that. And we are also not splitting data sets. What happened with us multiple times that we published a data set like a community, a single cemetery, and they like nitpicked the individuals that fitted their ideas. So basically they weren't treated as like whole communities. They just created subgroups inside them and started to use them instead of the whole data set, for example, which in a way like really hides the heterogeneity that are in some communities, and I think really creates like somewhat a biased analysis.
Thank you, István. Hannah?
Well, thanks, Jan. If I could jump in really quickly, this is so wonderful that we're talking about this. And I also want to just direct people towards a resource that was put together and published earlier this year by the National Academy of Sciences, Engineering, and Medicine. And they brought together genomicists, population geneticists, anthropologists, sociologists, all to talk about exactly this, about the title of this document is Using Population Descriptors in Genetics and Genomics Research. And essentially it's aimed at how to use and develop terminology to work with modern data and modern biobanks, but it's over 200 pages and it's really thoughtful, and it's something that, I think ancient DNA researchers could learn a lot from too.
Thank you, Hannah. Krishna?
Yeah, just quickly mention this kind of somewhat what István was getting at. Like this naming thing is kind of curious. So, we, along with Patrick, published one of the earliest medieval population genetic papers, 2018, and we were very careful when we did that. We wrote that paper to never ever say the word Lombard genome or Lombard samples. However, since that paper, you go into all sorts of databases and they have been relabeled as Lombard. This is sometimes by academics, but also by kind of amateur kind of genealogists, people that are making companies that claim you can get Lombard DNA. So this is something that happens quite a lot, and it's not just within academia, it happens outside. People kind of look at the paper with how we've written it and project their own ideas upon what these things are. So, and this happens frequently. I'm constantly reviewing a paper, and I probably give away who's the reviewer by saying, well, these are not Lombard DNAs. The authors never said these were Lombard, and I think that usually gives away that I'm the reviewer of that paper. But it's a constant thing. It's a constant battle, I would say.
Thank you, Krishna. Brian?
Yeah, regarding that, Krishna, are the projects who are involved in this sort of research, are they putting out sort of the equivalent of public service announcements or whatever the case may be, encouraging people to not do just that, to not talk about Lombard DNA? Is anything like this being made clear in these projects?
I can't speak to any of these other projects. I mean, we make it clear in the paper, but we have somewhat limited influence on what happens once that data is public.
Yes. So while we're on this question of how do we synthesize the paleogenetic and the historical data, or what our narrative sources say, so let me just ask another question. It comes from me as a historian who works primarily with written sources. And I'm curious, would paleoscience and archaeology, even based on their own resources alone, have ever suspected that such things as Huns and Goths and Vandals and Romans had ever existed? What could these sciences tell us about the history of this period without the written sources, just based on their own resources? Janet, can you start us off here?
That's a super important question. And I think it's important to recognize we're dealing with three incredibly different data sets. We've got, on the one hand, the labels, the Huns, the Vandals, and the Romans, which are lived social or cultural or legal terms that people use to refer to themselves and other living people. And on the second hand, you have the paleogenomics, which can show at fundamentally how large groups or small groups or individuals are biologically related to each other. And then the last set is the archaeology, which we assume has something to do with lived identities. And that's the assumption the field is based on to a large extent, the funerary archaeology. But at the end of the day, all we really know about what goes in the ground with a deceased person are the things that the people who buried them thought was appropriate for one reason or another, and we may never be able to recover exactly those reasons. What they thought those objects belonged with that person for something. And the trick going forward is to take an even more nuanced approach to figuring out how they all relate to each other. And it's a reasonable assumption that they all do relate to each other somehow, and that's what the field of funerary archaeology has been doing for the last 200 years. But at the end of the day, it's the places where they don't necessarily overlap that I think are going to cause the most difficult interpretative problems, but also the places where we can really strive to improve how we understand those connections from any of those disciplinary angles. So that's the short version, but I think there's a lot to be explored, especially in the relationship between biological relatedness, what goes into the grave of people who are biologically related. We might not be seeing their actual lived social kinship experiences, but whether or not those objects correspond to any particular ethnic label, or in the case of the Romans, the legal label, I think that's one of the big questions for paleogenomics interdisciplinary projects down the road.
Thank you, Janet. István?
Really, just to build upon what Janet has said. So I think written sources give us names that we use as labels, but also a framework that shape our thinking and ideas about certain periods. It's not true to all periods. Sometimes you don't have written sources at all, but in all periods, it definitely gives you a framework that you are thinking about. But even without without any written sources, we would be still looking at groups. Just look at the archaeology of prehistory, for example. We are still looking at groups, and why is that? Humans are social creatures. We form groups, we form communities, even larger groups, etc. And being part of a group and representing your belonging to a group, I think is important in everyday life. And this is what we are looking for in archaeology as well. So basically, burial archaeology is looking for patterns in cemeteries between sites, etc., and compares these patterns and tries to identify groups inside sites, but also groups between sites as well. And we describe these. Basically, that's the basic archaeological method. And there are clear differences between chronological periods and smaller or larger regions. For example, it's very easy to differentiate between a 6th century site in Western Hungary, the former territory of the Pannonian provinces, and the Tisza region, which is east from the Danube, so would be in the former Barbaricum, for example. They differ in terms of grave design, they contain different artifact types, etc. So even if we wouldn't have the names, let's say Langobards and Japids, we would still think that they belong to different groups somewhat. I think the problem really comes when we try to identify like typical burials or typical types, and then describe whole communities based on these identified typical like versions. And then based on these like homogenized picture, we start to compare them on an individual basis. I think that's there where really the problem starts, because this leads to a very like distorted image that hides the heterogeneity of these sites and communities. And it also hides the many different reasons. One of them could be ethnicity, but could be a lot of others as well, why they could be different or similar. So I would say that even without textual sources, written sources, we will be still speaking about groups, because that's what we can see. But yeah, textual sources might give you a framework that could like distort your ideas or like how you understand the data. Thank you.
Thank you, István. Krishna?
Yeah, I mean, from perspective, if we were to just look at the genetic data, we didn't have any knowledge about history, which many geneticists probably don't anyway, then it would be interesting to see how we'd interpret that data. I think we're only now building a lot of genomes from the period that we're talking about here, kind of late antiquity, early medieval. I will say you would see different groups emerging. But you'd also see them quite cosmopolitan cemeteries, for example. You see lots of different genetic types or ancestries in there, and it'd be interesting to know how some people would interpret that. But you also we're starting to see clear evidence of big migration shifts in different kind of periods. I don't think we would be able to say, okay, these are, with such definition, these are Huns or Goths or Vandals or things like that. But we would see groups emerging. There's kind of a geographical structure to it, but then there are these kind of clear differences between regions and clear incomings and outgoings, often strong signals. So the historical data is absolutely necessary to kind of interpret that, the framework, as István said. But it's not like it's just a random mess and we wouldn't have come out with any patterns from the genetic data. There is things there, there is structures there, and it changes over time that suggests movements and mixing as well.
Thank you, Krishna. Patrick, you'd raise your hand?
Yes, just to continue along exactly the same lines that István and Krishna have been discussing from our own work, when you ask about what happens when we add these historical labels to the mix, we're finding in our work we have clusters based on material culture. We have clusters based on genomic ancestry, but these do not necessarily overlap. And we find an incredible variety of genetic backgrounds which may share the same material culture. Not always, but sometimes. So probably, as Krishna talked about, cosmopolitan, and I think that these ethnic labels that we see in our late antique texts really need to be understood as something other than what we today understand as ethnic, and think about these more as political, perhaps, constitutional, in which there may be a core of people who intermarry a lot and thus they have a genetic profile. But there are a lot of other people too who are part of that world, who are sharing the same material culture, presumably the same values, and the label that the historical text would give them might not at all correspond to any genetic grouping or any archaeological grouping. So again, these are just very different things, as Janet said, and it becomes a real problem when one tries to move from these textually recorded labels to either biological or material culture groupings.
Thank you, Patrick. Brian?
Yeah, I very much agree with what's been said here, given the cautiousness that we need to have when associating genetic findings with ethnic names. Though when I'm reading, maybe this is just I'm not quite putting two and two together here because I'm not intimately involved with this research. But like, for example, a recent study about the Avar elites and burials and genetics. It seems that when these studies are published, there's no problem, or y'all have no problem, or these groups have no problem calling these Avars just outright. Is that just a matter of for simplicity, or is there some other methodology underlying your confidence in calling these people Avars?
István?
I think we are calling them all Avar period elites. I think there is a distinction there. Definitely, there is definitely a distinction in how you think about it. So Avar period is a term that's used similarly, let's say, early medieval period. It really like describes certain centuries in the history of a region, in this case, the Carpathian Basin. This doesn't mean then that everyone is an Avar. It's basically a term that helps you orient in the history. Like just saying centuries doesn't really tell much. Of course, for a lot of people, also Avar period doesn't really tell much, but that's basically a chronological indicator. And we are speaking in that paper, about elites that lived during that time. I think there is a distinction there.
I think Patrick might have something more directly relevant to this line.
Well, I was simply going to say, as István says, that I think the article that you're talking about is done by some people who are in our group, but it is not our. I don't think you mean the Cell article on migration from Asia, but maybe you do. We have an empire that is an Avar empire. This is a label that is apparently used by these people according to contemporary sources. And within that empire, there is, based on material culture, an elite group within that empire. So when we talk about Avar elites, I think that what this means is elite individuals who presumably have access to power and resources within this polity. It is a statement about their political status within an empire. It's not, in the first instance, a statement about their genetic status. When we begin to look at these people and we look at their genetic background, then it becomes very interesting to see how closely related they are. We also find it very interesting to see that these individuals have very close relations to Eastern steppe populations, quite far into Eastern Asia. And thus there is a community here. Are they the only Avars? No. Are all of them Avars? Who knows? But they are a group that probably, as in many cases, by marriage alliances and maintaining these kinds of connections, maintains a political position within what is definitely a polyethnic empire. And in that sense, I think it's fair to call them the Avar elite, or if you want, the elite of the Avar empire.
Thank you.
Thank you, Patrick. Hannah, you had something.
Awesome, thanks. Yeah, I was just going to sort of mention a point made by Cyprian Broodbank in Making of the Middle Sea that I find really helpful in thinking about sort of what we can and can't know from any given line of evidence, including genetic evidence. And I'm going to read it so I get his quote right. But he says, without delaying the likelihood of various constellations of social, cultural, and other identities, early Mediterranean history is instead comprised of an ever-shifting kaleidoscope of webs of peoples and practices changing within and between places. And I really like that sort of, it reminds me that people have multiple identities, multiple ways of seeing themselves. And so with genetics, we can learn, things about ancient people and cultures, but we should never treat it as essentializing, right? That there's so many different ways that somebody would describe themselves, and we should be mindful to think of the people we study as that nuanced as well.
Excellent. Thank you. I will move now to our next question, which is on narratives of migration. So narratives of migration and ethnicity are traditionally rooted in written sources. How has scholarly thinking about these narratives changed in recent times? And has paleogenetic analysis tended to support, nuance, or refute these narratives? And have you ever been surprised by what emergent paleogenetic data implies about existing historical narratives and debates? Brian, let's start with you.
Sure, because I'm not intimately involved in these projects at all, I can take the first part of that question, and then I can pass it off to my fellow discussants. I would start by first saying that our understanding of barbarian migrations, whether they happened or didn't happen, our understanding of barbarian ethnicities, whether they were real or just an illusion, are indeed primarily rooted in the literary record, in written sources. So, migrations. The sources that were written during the so-called migration period usually don't give us continuous accounts from beginning to end. We instead use narratives that are partial, that are discontiguous, and we have to stitch them together. Now, the act of stitching them together, or the refusal to do so by some, that's a matter of art, and then therefore debate. So, of course, think of those infamous maps with the arrows showing barbarian migration paths. So the Goths, for example, would go from the Danube down to the Balkans, to Italy, and finally to Southwest Gaul. Note how that arrow, though, maintains like a similar shape. It maintains a similar color. It cuts right through the Romans. It walks right over the Romans. It arrives in Gaul basically unchanged. Now, those arrows, I would say, they're the product. They're a visualized argument of the stitching together of those sources. Now, I don't think there's anything inherently wrong with stitching together sources. Like we kind of have to do that. The problem is sort of stitching together sources and, I don't know, like turning them into a sock puppet that ends up mouthing modern assumptions rather than the realities of the past. And so those arrows end up representing old understandings of not just migration paths, but also old understandings about who these people were, how they were constituted, as being, homogeneous, distinct from the Romans and their bloodlines, so forth. Anyway, today's scholars have very different understandings about these processes, but there's still a lot of debate. So the Goths, again, no scholar would suggest that the Goths ended up in Gaul entirely unchanged after hanging out inside the Roman Empire for 50 years. But there are some scholars who would argue that the Goths who do end up in Gaul basically had nothing to do with the Goths who crossed the Danube years earlier. The important implication there being that there was no migration at all, that these were just different groups who had the same name at different times, and then instead the foundation of the Gothic kingdom in Gaul is based on the contingencies of Roman politics, warfare, so on and so forth. So this isn't the story of a people, of an ethnic group. Instead, this is probably an army whose warriors were essentially themselves Romans because they had been interacting with Romans for so long. So that's sort of one group of scholars. There are others who would disagree with those models as being overly skeptical, that sort of run the risk of overlooking important historical processes like migration, or even of erasing entire peoples from history by denying the reality of their ethnicities. So these other scholars are more open to accepting large-scale migrations and to understanding ethnicity as being, at least in part, an important factor in group formation there. But regarding ethnicity in particular, I should just say this. All sides of these debates now agree with this kind of post-World War II understanding that ethnicity is fundamentally social and cultural. It's constructed socially and culturally. It's rooted not first in genetics and bloodlines. Peoplehood emerges when people agree that there are people, not because of physics or whatever else. So with this in mind, barbarian historians reapproached the sources. They ended up finding discontinuities in the histories of barbarian groups. They found out that they were heterogeneous. A Gothic group might contain some Huns and some runaway Roman slaves or so forth. And so this whole idea about genetic ethnicity is just right off the table. But even so, there are still legitimate arguments being made that barbarian ethnicities were still socially operative forces. They could be durable. They could provide groups with solidarity, cohesiveness in hard times like migration. Whereas more skeptical types tend to think that ethnicities were threadbare, they're presented as so rhetorically wrought in our sources that we can't know one way or the other. Or even if they did exist, they were evanescent and could be easily kind of sloughed off if it was in their advantage to do so. So that's more or less the lay of the land regarding migration and ethnicity as I see it.
Thank you, Brian. Patrick?
Yes, I think that Brian has very well summarized the state of the debate about ethnicity, migration, and so forth from historical sources. It's become a polemicized field, as so many things have, and many of the most vocal actually are arguing about very fine differences in their interpretation, I would say. But apart from that, I think that what paleogenetics brings to the table can be helpful to look for a middle way between those who say, oh, there are no migrations, and those who say there is continuity. Well, we're not looking at Goths, but at this Avar kingdom, we have a poor population with very strong connection to the Eastern Asian steppe from the early 6th century, who appear and create a new polyethnic empire between the Vienna woods and the Carpathian Mountains. Was there a migration? Absolutely. Are they all pure something? Absolutely not. There are already mixture. They pick up additional genetic components as they come through the Pontic steppe. But this is a significant change within the genomic makeup of Western Europe that was simply not there in the 5th century. This is there a change? Yes. In terms of the written text, actually, this is a fun case. We have two competing arguments from contemporary authors. One is that these Avars are the remnants of the Rourand Empire that is destroyed by New Turkish Empire in Eastern Asia, and they move west. Then we have, according to Byzantine emissaries or Roman emissaries to the Turks, oh no, the Turks say these are just rebels from our Western steppe people who run away, and they take the name Avar because it scares people, because it's a famous name. So we have two competing stories. These are pseudo-Avars, or they really are people from Eastern Asia. Well, they really are people from Eastern Asia. That shows that. Are they the only Avars? Are they a pure something or another? No. Does the meaning of Avar culturally stay the same over this through a process of migration, creation of a new empire and so forth? Certainly not. Does it change over the course of the later 6th, 7th, and 8th century? Certainly it changes, but we can see movement. We can find communities that maintain a very strong Asian genetic identity over 7 generations in what is today Eastern Hungary, Eastern Austria, finding wives that come from someplace who have the same background that they do. Is this ethnicity, or is this a clan? Is this a tradition that we marry those people? We may all be Avars, but our group marries people like this. We needn't jump immediately to big ethnic labels, which I would again say are, to a great extent, political labels. They are constitutional terminology, even though there is a tendency to interpret them as some kind of unified value. In the world of the Langobard invasion of Italy, our sources tell us that this army, it's the Langobard army, but it's made up of all kinds of people, including Romans, Gepids, Sarmatians, all sorts of other sub-labels who somehow or another are at the same time part of a Lombard army. We see a genetic change in Italy, but it doesn't mean that all of these people who have a new genetic background from the indigenous population would immediately be considered in all circumstances Lombards. They might be part of the Lombard army. They may have an identity that is something else, maybe Thuringian. Maybe their primary identity is with their family or their relationship with a political leader. We're told that the Duke of Turin is a Thuringian. He's a Lombard Duke who's a Thuringian. What does that mean? So I think that we can see a genetic change, we can see real migrations, but it does not mean that these big overarching labels can be applied to all of these people in all circumstances, or that the labels continue to mean the same thing before, during, and after these population movements.
Thank you, Patrick. István.
So from an archaeological point of view, I think it's very interesting to see this like paleogenomic data coming in, because archaeological interpretation like really oscillates between migrations and like local developments. For decades, everything was interpreted through migrations at every change, every shift, and then it really moved away from that, and everything was interpreted through like local development, etc. And in a way, like these new data really like led to like a resurgence of migrations of like as an interpretation in archaeological research, but it's not like really a one-way thing, because it doesn't exclude or reject the local development theories. In sometimes it really goes side by side. In some cases, it actually opens up doors that we like didn't really consider, or possibilities that we didn't consider before. Like the Langobardian migration in the 6th century from, let's say, today's Czechia through Pannonia to Italy is one of the like probably best attested migrations in written sources, and also archaeologically. It was never really like questioned in a way than many other migrations have been. And for example, in the 6th century Solad, which belongs to this Langobardian migration, presumably is actually never considered as a result of local development. But in studying multiple 5th century sites around the vicinity, we have found out that the genetic variation, the genetic background was already present there in the 5th century. So it actually doesn't have to be people coming in. It could be a local development as well. It of course doesn't mean that they are not like new migrants in the territory. It just really shows that it actually can like swing both ways, I think, this new data, and that we really have to go like side by side, region by region, and take a really like nuanced look.
Thank you, István. How can scholars responsibly draw connections between biological relatedness and genetic ancestry on the one hand, and social kinship on the other? So when designing a research project, what are best practices for avoiding the pitfalls of the biology of race? For example, there has been a tendency recently for narrowly focused micro-studies. I mentioned the recent Lombard, Anglo-Saxon, Avar studies. What do we gain and what do we lose by focusing tightly on subpopulations within a cemetery? István?
These are a lot of questions, and these are questions that I would be able to talk like hours about. So first of all, I don't think that any of the currently running projects actually like seek to illuminate the connection between genetic variation and cultural difference. I think that's a huge question. I might be just too young to like really try to like understand it or like answer a question like that. I think there is a clear tendency towards smaller, more focused questions in these papers. And fortunately, many of these questions are actually formulated by historians and archaeologists. Many of these questions are already present in historical and archaeological research for decades or maybe even centuries, like the already mentioned origin of the Avars or origin of the Avar elite. And in most cases, of course, written sources, as Patrick pointed out, are ambiguous at best, and archaeology also developed multiple theories based on the same exact data set. Sometimes these theories are like quite the opposite, but that's really the nature of archaeology. And I think like the results of these more focused studies also provide valuable information about these much larger questions, but more like a side benefit, not as a main goal. And I think we need a lot of like these focus studies to actually be able to start answering huge questions like that. In regard of like the second part of the set of questions, so why are these, what's the benefit of these more focused studies? I think like this tendency towards like these studies and also this new approach of complete sampling of sites means that we are really moving away from generalizations and moving towards like a better understanding of communities and extra and inter-regional variations that we've already talked about a bit. Like in the already mentioned study of the 5th century Lake Balaton sites, we studied 3 sites. They have a slight difference in terms of chronology. All of them are in a very small area of just 200 square kilometers. And yet what we have found is there are like significant differences in terms of genetic background, but also in the structure of biological relatedness, and also what's its impact on burial customs. And this, I think, really leads to like the third part, like what's the relationship between biological relatedness and social kinship. That's a huge topic in itself. But I think, so from an archaeological point of view, first of all, that's something that I was interested in as soon as I started to write my master's thesis. That was about kinship, but purely based on archaeology. Archaeology was always interested in social kinship. Sites has been described as family cemeteries, or burial groups has been described as family groups for like really decades, or from the beginning of archaeology. Multiple burials, especially if there is like an adult and a child buried together, were identified as signs of kinship. For example, in a 6th century cemetery at the Tisa region, we have 2 young children who were buried with the different halves of the same pendant. And of course, it already raises the questions, are they siblings? Are they connected somehow? So we always had these questions, and we try to identify subgroups inside communities to like really understand their organization. And now we have another set of data that we can involve in these questions that's provided by paleogenomics. And of course, our main question in regard of like biological relatedness, so are these connections expressed in the burial customs in any formal way that would suggest that these connections were actually known and acted upon by the community? Are related individuals, I don't know, buried together? Do they form separate clusters in the site? Are there similarities in grave design or in artifact types? These are the things that we are looking for. And if biologically related individuals show connections in the archaeological data, then that could be understood as a sign of, let's say, social ties between them. Isotopic information about diet and mobility, like sharing similar strontium values, let's say, could also be a good indicator. In most cases, like based on our like results or what we have seen so far in the project, we see that biologically related individuals actually tend to be buried together. In the communities that we have investigated so far, they are buried together, or in some cases, the whole communities are actually organized around large extended pedigrees. But we also have cases where there is no clear connection, like basically no connection at all between biologically related individuals. This still doesn't mean that these are like not important socially, but still like really like shows a different kind of perception in the community. And I also think that, first of all, yes, social kinship is not the same as biological relatedness, but in a way, biological relatedness could be a strong basis in most communities of like social kinship. But indirectly, I think genetic data also provides us information about social ties that are not based on biological relatedness. For example, we have whole burial groups where everyone is connected biologically, but there are few outlier individuals who are not, still buried with the same group, have the exact same burial customs, etc. So they definitely belong to the same group, yet they are not connected biologically. This really suggests that, or shows that you can connect to these groups multiple ways. Just think about marriage, or like in a more natural forms, like reproductive partners, which is like a very important social connections in most communities, but would be invisible genetically. And we have a high variation of these types of connections. But what we have found so far, that biological relatedness in most communities in this period, in this region that we are studying, actually is a very strong basis for social kinship in many ways.
Thank you, István. Janet?
Along with István, will preface this. This is the short version of what is usually a 45-minute lecture. But I think that last sentence István just said with very careful time, manner, place descriptors, trying to responsibly draw connections between biological relatedness and genetic ancestry and social kinship. I think we're well on our way, but I think it's going to come down to a really careful use of language between and among different research groups, but especially also in public-facing publications, because for a number of reasons, there are just disconnects between how we use different words to mean different things. The one that most easily comes to mind when talking to the public is textual historians use ethnicity to study the early medieval period very differently than, say, modern American culture uses the word ethnicity, which is very often what is perceived to be a politically correct way of saying race without actually saying race. And there's also this kind of popular perceived notion in especially modern DNA testing kits, where you send in a sample to a company and they tell you you have such and such % of such modern geographical DNA. There's a really, I don't know if famous is the right word. It's famous to a certain small subgroup of academics. But there's a commercial that Ancestry DNA put out 5-6 years ago now, maybe, about this man named Kyle who grows up in a very German American cultural structure. And he apparently famously wears lederhosen, which seems one of the more improbable aspects of that commercial. But he sends in his testing kit, and Ancestry DNA tells him that he actually has more Scottish DNA than German DNA. And so I think the line is he, quote, trades in his lederhosen for a kilt. So his entire lived cultural experience is thrown out the window because some large genomic company told him something different. And that is such a skewed notion to sell to the public about the relationship between genetics and culture. Culture is not biological. And I think that's an obstacle that has to be overcome just on a popular science level, as well as in the academic publications, because, as Krishna was saying earlier, Lombard DNA is not an accurate term. And I think the nicest way to say it, Krishna, might be is it's a popular shorthand for what otherwise might be a really clunky, but much more accurate set of words to describe what we see with biological relatedness. And this comes to the foreground a lot. I work on late Roman and early medieval Britain, and there's this question of when can we call people Anglo-Saxon in the historical past and have it mean something productive? For example, there was a recent paper in 2022 by Geary et al. About Anglo-Saxon DNA. And I think using those cultural words as a shorthand is partly driven by publication. I mean, we have word counts. We have to say things quickly and concisely and somewhat elegantly. And it's really clunky to say every single time people who lived in 5th or 6th century Britain, which is much more accurate, but that's a large word count when you have to say that 20 or 30 times in one page. So I think we have to figure out what language we can use and what language we communicate with the public, but we cannot wind up back in the 1930s racial science idea of that culture is biology, which is just abjectly incorrect. So I think going forward, we can draw these connections between social groups and social kinship and relatedness and perhaps even ethnic labels if we can get solid enough evidence for all 3 points in one place. But the language is going to be important. The way we communicate that is going to be, I think, most important of all.
Thank you, Janet. Krishna?
Just going back to this idea of designing a project, and that these recent ones are narrowly focused. To give a bit of context, this Lombard project, Patrick and I have been working on this for 15 years. It's not a recent thing. Now the technology has caught up that has allowed us to now expand the scope of these projects. And I certainly wouldn't consider the Avar project to be micro. That's a huge, huge region and a huge kind of time period that's being covered. So these kind of focus on the micro studies is somewhat a limitation of technology at the time, right? So the idea of doing the Lombard project, Patrick will remember better than I, comes from Patrick's own seminar in the history department, and it's actually designed by historians, and I was just there to and other geneticists were there to give advice of how genetics could help develop that question. So it's not a question that's actually emerged from geneticists. And over time, it's grown into these new things that we can do, these big scale periods of time. And now the AVAR project is not a micro study. It's covering so many dimensions, periods, regional, within regional things. And that is only going to get more and more prominent as sequencing capacity goes up and things get cheaper. Like doing 6,000 genomes 5 years ago was unthinkable, completely unthinkable, not possible.
Thank you, Krishna. Patrick?
Thank you, Krishna, for giving us that background to both of those projects. I think that there are two issues that have been discussed, and I simply wanted to underline them in this section. And the first is that we're doing a lot more in paleogenomic studies than looking at migration. And one of the most interesting things to me is not did the XY migration take place, but how are local communities structured? We see with enormous detail the process by which a local community is formed, in some cases from a couple of founding individuals. We see the marriage or mating practices and structures. We have cases that in enormous genealogies over several centuries, and there is not a single case in which there is a marriage between two people who are within 6 degrees of relatedness. This tells us that people have a powerful knowledge of their own background. We see enormously strong patrilinear patterns in which these communities, not all of them, but many that we've studied thus far, with different cultural backgrounds, are very heavily the patrilocal. The women are coming in from the outside with very, very few exceptions. We are seeing such things as levirate marriages within the society in which a woman has a reproductive partners, a number of them who are related by as men, perhaps with a stepson, with a brother-in-law. Presumably the first husband has died. This is she is kept in the community by then marrying someone else. So we're seeing a lot about the structure of communities and how they change over the centuries, really getting into that. So this question about migration, ethnicity that people are obsessed about, I think because of our own obsession today about, as Jen had said, in America, race, in Europe, ethnicity, there's so much more to help us understand past communities that this paleogenomic analysis is making possible. That in a way, it's much more interesting to me to look at a community in northern Italy and how it develops and changes, how different groups with different backgrounds do or do not connect, than whether or not they came as lumbered. The other question about these micro studies is, this is a very new process, and we need to start at a detailed level with place and time at a specific level. As István says, we have this 200-kilometer area, and the genetic diversity represents everything from today, from Scandinavia to Italy. These are very, very complex communities that would not be seen if we didn't do these careful micro studies. And in our big histogenes project, we have a whole series of these, but that's only a first step. The next question is, at a regional and trans-regional basis, how do we tie these together? One of the techniques that István and Krishna can talk about is identity by descent, creating networks that show biological connections, tying communities together. with certain nodes that are very dense in connection and others that are not, and then relating those individual nodes to presumed social status based on grave goods, location, are they male, female? How do we see networks created? And then when we add to those networks, we overlay them with networks of culture, material culture, grave goods, eventually isotopes. So when you talk about these micro studies, yes, they're micro studies, but we're also doing transepts that will try to look at a region diachronically and see the transformations that take place from the Roman imperial period to the 10th century, but also tying together. But the first step is to understand comprehensive communities, not sampling in a cemetery, but sequencing everybody we can get out of that cemetery, looking at how they connect with each other, looking at the correlations between material culture, isotopes that tell us about diet, about origin, and all of this together. And then we move progressively beyond that through identity by descent networks and other means to expand this, to try to get a larger picture of the transformation of Eurasia at the end of the Roman Empire.
Thank you, Patrick. Brian?
So I want to pick up where you left off, Patrick, in focusing on the word you used, how. You want to describe how these people are related in terms of marriage here. In the Avar case, it really is quite striking that they're so deliberate in their choice of marriage partners. So it's good, I think, to right now be descriptive about it, to label these as sort of cultural practices, marriage practices. But I suspect, knowing that my fellow humans are curious, they'll start asking why, not just how they're related. And they might start asking, are these practices, do they bear on notions like group identity? Are these things inherently meaningful? My question, I guess, is, do you think it's inherently responsible to start asking those questions, like why? Why are they doing this? What might it mean to them? Or is this kind of work purely descriptive as of now?
I think that what we're trying to do at this point is to describe. And no amount of detailed genetic analysis is going to tell us why. It's not going to tell us why people move from one place to another. It's not going to tell us why they created reproductive units with this particular man or this particular woman. So we could sequence the living daylights out of all of our people, and we're not going to answer any why questions. But of course, history has always been about describing and explaining. I mean, if we go back to Herodotus, he wants to understand what happened between the Greeks and the Persians and why this came about. So the why question is certainly there. The danger is to jump to the why question too soon. But of course, we want to understand these things, and this becomes interpretive. And these people who say, oh, now, thanks to genomic data, we will definitively answer all of these questions, is hubris or ignorance. Once we get into the area of interpretation, then this is going to be a question of our subjective attempts as best we can to explain these. We look at modern patterns, for example, with levirate marriages in steppe populations in Mongolia in the 19th and early 20th century and say, aha, this is a practice and it is done in order to maintain the dowry that the woman would bring. And so this is why they're doing this. Well, maybe, maybe not. The interpretation is certainly appropriate. What I don't like to see is when we do a very careful study, like István said, of a couple of Lombard area cemeteries, people jump in and say, aha, that proves that the Lombards marched from Scandinavia to Italy as a pure race of people, and I am a Lombard. That kind of thing really upsets me. But yes, the why question must always be there for us historians, but let's find out the what. And then we can debate the why.
Thank you, Patrick. István, you have something to add to this?
Yeah, just a tiny bit of information that it still might be missing. So when we are speaking about like correlation, biological relatedness, and social kinship, we are speaking about like first, second, third degree relationships. But paleogenetics has the strength to identify these like 7th, 8th degree relationships as well, which might not mean anything socially. It really just shows whether like different groups, communities were actually pulled from the same population pool or different one, how they are tied together. So there are clear differences of evidence that we can look at.
Thank you, István. So I have a relatively technical question here, which is how are recovered human paleogenomes dated? Tim, who's watching here, not speaking, it reminded us in a conversation we had, a planning session that we had a few days ago, that in pathogen paleogenetics, one can molecularly date genomes. Is that possible for human paleogenetics too? Are multiple lines of evidence leveraged for a more robust dating? Or how much of the evidence is ultimately based on traditional archaeological means of dating graves, potsherds and other material goods that are buried, which sometimes have a considerable range. They can sometimes be a century off for all we know, and that can significantly affect how we use paleogenetic data, right, in reconstructing human populations. István, you want to start us off with that?
Yeah, sure. So I think it really depends on the period. In late antiquity and during the early medieval period, which is, let's say, from the late 4th century up until the 8th, 9th century, which is the main foci of the Histogenes project. So archaeology has developed chronological frameworks based on stratigraphy, coins, typology of various artifact types, and so on. And we support these archaeological chronologies with radiocarbon dating, of course. But archaeological chronology in most cases provides a more accurate dating due to issues with like the calibration curve in certain centuries, for example. But there is a clear limit to it. First of all, we can of course only date graves where we have like grave goods or any kind of like datable artifacts or phenomena. And we usually can date only like a limited or a smaller number of graves in each site, so not every one of them. The combination of the like already mentioned methods and the pedigrees that are provided by paleogenomics means that we can date more graves and refine the internal chronologies of the sites like even more. In some cases, we have seen changes in the chronology of sites, but this is like really present in periods like, let's say, late Roman period or the 9th, 8th century of certain regions, when we have a large number of burials without any kind of datable artifacts. In generally, our experience is that archaeological chronology holds up really well in most of the cases that we are studying. But this is specific to this period and to this region, of course.
Thank you, István. Hannah?
Yeah, thanks. And this is a really great question. It was really thought-provoking, actually. And just a little bit of the background on why we can do this kind of dating on pathogens is that many pathogens, and maybe for this discussion, we can take Mycobacterium tuberculosis, which is the bacteria that causes tuberculosis as an example. Lots of pathogens reproduce clonally, which means that an offspring is almost identical to its parent, other than mutations that occur. And that's, of course, different for people. People reproduce sexually, which means more or less 50% of your genome comes from each of your biological parents. So there's sort of that difference that allows us to use this molecular clock with pathogens and not in humans. I bet people have tried, but these mutations are really informative, and they can be informative about selection on certain traits. For instance, each of us has about 40 to 80 of these new variants that neither of our parents had. These are just new mutations that have occurred in the genome. A lot of them don't have any measurable impact. A lot of them probably don't do anything. Some of them, may cause some change or some different trait. There's a paper from 2015 that essentially looked at these. They measured what's called a singleton density score. That's when a singleton essentially refers to when one of these new variants is so rare it's only been sequenced in one person ever. But essentially they were able to look at different traits in the UK Biobank and see that certain traits like lactose tolerance were selected for very recently. And sort of what they did is they would say a low density of these means it was this trait evolved and was selected for recently. So they said the past 3,000 to 4,000 years. And that if they had seen a higher density of singletons, maybe that would have indicated that the selection was earlier, so maybe around the Neolithic. And so that was really interesting because that was before there were a lot of ancient genomes published. And then it's been sort of ground truth with ancient genomes. So it's kind of cool that especially in Northern Europe, this trait has been selected only within the last 3,000 or 4,000 years and not the last, 8,000 to 10,000 years or something like that. Yeah, so essentially that might be a way that you could get information about the timing of selection or the timing of a variant from these mutations, but I wouldn't use it to date. I wouldn't take a genome and say, oh, look, I see this variant. I'm going to say they're from, the Roman period. I wouldn't do that. So I would definitely use radiocarbon dating and contextual information as much as possible when you're thinking about when an individual actually lived. Yeah, thanks.
Thank you, Hannah. Krishna.
Yeah, a quick note on that. The function of being able to take a genome and dating it from its mutations will increase the more genomes are sequenced. The more genomes we get, the easier it will be to go, oh, this genome should be this years old based on all the other genomes that exist, because you can contextualize genetic variation. So that's a kind of important. And also, it will come from doing whole genome sequencing. When we say we're sequencing genomes, we're not actually often sequencing whole genomes. We're sequencing quite a lot of the genome, but there are groups that do a lot of whole genome sequencing. And when you do that, you'll actually start to get those kind of rare singleton mutations that Hannah's talking about. And then you actually will increasingly be able to date an individual purely based on their genome within the framework above. That is something that will be increasingly possible, and the confidence intervals will get narrower and narrower the more genomes there are.
Thank you, Krishna. István.
I just wanted to add that time frame is very important, and it's very different in different periods. Like, for example, radiocarbon dating gives you, let's say, 80 years, like a range of 80 years, which is amazing in certain periods. But in our period, it's almost useless. Like, we have to go down as far as we can, like really pushing the limits with the combination of different methods, because sometimes like 50 years, that's even like too wide of a range. So there are different needs in different periods and in different projects.
Thank you. Hannah, actually, I have a question for you because migration has come up often in the discussion. And it came up when we were talking about the narrative sources. And then Patrick rightly reminded us that all of this research is not just about migration, however much it may tend to fascinate, the general public and so on. But earlier when we were planning this conversation, you made some very interesting points about how we think about migration that I think our audience should hear, because there's some important clarifications and distinctions there that you made. Can you tell us a little bit about that? So how does your research tie in with the study of human migration, mobility, and what may be missing from this area of research?
Yeah, absolutely. And thanks so much for asking this. I think that migration has been a topic of ancient DNA studies and sort of used, as Patrick was saying, as a conceptual explanatory mechanism for a lot of genetic changes that we've seen, maybe at the expense of discussing or even considering or thinking about other types of mobility. And I think this happens for a couple, and I'll get to those in just a second. I think this happens for a couple of reasons. As one, that migration, and when we're saying that, maybe, when in humans, we're talking about sort of a unidirectional and one-way movement. It's easier to observe in genetic data. And then the other is that in population genetics, migration actually often refers to a broader range of mobility patterns than when we're talking about humans. So when you think about, caribou migrating south for the winter or something like that, for humans, we might call that seasonal mobility. So I think in part, it's just been different fields having different definitions of this term, but part of the result of that is that it's led to papers sort of using migration as an explanatory mechanism rather than things like seasonal mobility, state organized mobility, forced migration and enslaved persons being forced to move, pilgrimage, tourism, things like that. So there's sort of all these reasons that people move. It may be one way, it may be multiple directions. Patrick mentioned exogamy and marriage patterns that people might be moving for something like that. But I don't think we would want to use the term migration for that either. I guess my thought was just that, the social sciences have developed this really rich diversity of terms and concepts to describe human movement. And it's exciting to see more and more people sort of start to draw on this full range of options when we're talking about people in the past. And one example that I really like is from Mark Haber's team, where they're looking at people who they think were crusaders in the Eastern Mediterranean. And so we wouldn't want to say that as migration, right? That these were people who maybe moved for a very specific reason and died there. So we find their genomes there. Yeah, but just to make sure that we're capturing the nuances of how people are moving. And I think also, and I don't know if we'll have time to discuss this or not, but I think being careful about the language we use also sort of can prevent our work from being used in ways that we might not intend to. That sort of the more clear and accurate we can be with the terms we use, the more we prevent it from being co-opted for reasons that we didn't intend for it to be used for. Yeah, I don't know if others have thoughts on that.
Thank you, Hannah. Patrick.
Yes, very briefly, I would certainly agree with Hannah, and I think we have to also think about distinguishing migration. We have to talk about a temporal element. If a population change takes place over 1,000 years, is that a migration or is that a diffusion? We have to talk, is a migration if a population moves 50 miles, 100 miles, 150? When do we start getting to a migration rather than simply population mixture? So I think that there are a lot of problems with the term migration, and there are certainly early medieval historians who would prefer to talk about diffusion of populations rather than migration. There's also chain migration. There are reverse migrations in which people move back and forth, and it would be very surprising if we did not see that same phenomenon. So all of this needs to be unpacked. And again, as in much of this discussion, it's a question of language and how we find the words that we need to use to express precisely what we're talking about.
Thank you, Patrick. Well, this has been a fascinating conversation, and it's time to bring it to a close. And I want to ask a question about what comes next in the study of interdisciplinary human paleogenetics, and what has to be done, what opportunities remain, and for late antiquity specifically. So I'll ask Patrick first if you want to weigh in on this, and then we'll see who else has some comments to make. Patrick.
Well, there are some very basic things, simple but to state, but hard to do. We need more coverage. I mentioned that France is a black hole for reasons that have to do with the direction of research in France, the funding and issues. We simply do not have late Roman and early medieval genomes or from what is today France, a very, very vanishingly few of those. We need to fill some of these holes. We have much more from Central Europe than we do from Central Asia and certainly East Asia, and there are political issues involved in that. African ancient genomes are beginning to appear, but so there's a problem of filling in a lot of blanks on the map. The other question is, how do we get beyond the tools? Krishna had pointed out that we get a lot of genomic data, but we don't actually use it all. One of our most common procedures is using principal component analysis, which is a radical simplification of the genomic data. And we need to figure out how to tease out more information rather than throwing it away because of the computational problems involved in this. He also mentioned that many of the studies, including ours, when I say we're doing 6,000 genomes, these are actually about 1.2 million points on the human genome that we have accessed. We're not doing the 2 billion plus sites on the genome, or is it 3 billion? So we need to think about what we can do if we really had whole genomes. So there are a lot of technical questions here, and there are methodologies. And then we also have to get beyond the old questions, trying to answer 19th century questions with 21st century methodologies. We need to develop new questions that our data allow us to ask that could never have been asked because from textual or material culture evidence. But if we just keep trying to answer the questions that people asked in the 19th century, we're really not going to make much progress.
Thank you, Patrick. Janet.
I feel like all of my comments so far have been ways that we have to be cautious and things we can't do. And I don't mean to imply that's what this field is. I'm actually really excited by what paleogenomics can do in conjunction with the archaeology and the anthropology and the textual history. And I absolutely agree with what Patrick just said. We can't answer all of the questions we're used to, but we can certainly find new things we weren't expecting and trying to explain them. We can ask new questions. We can break out of those old pattern molds. And that's really exciting. And the new data, the bigger the data sets we get, the more questions we can try to figure out, the more patterns we can see that we could not have even anticipated. So I think that's going to be a really exciting future for paleogenomics. I do think it has to be in conjunction, and this is where everyone gets to join in this really exciting new research direction. It does have to be, as Histogenes is doing, an interdisciplinary conversation, not where any one particular discipline is driving the work or the questions, but where everyone gets to come to the table and contribute to that conversation about human relatedness and migration in the past and how we might be able to look at it differently.
Thank you, Janet. István?
When I started to work with burials and cemeteries, I had no idea that one day someone will be able to tell me that this individual is the parent of this individual and what type of connections are between them. That was pure science fiction. And then we started to talk with Patrick and Krishna, and I think it was 12 years ago or something like that. And it became like a reality. This is now we are seeing a lot of cemeteries, but I think there are still some things remain. But of course, this is like the land of my dreams. Like they are extracting DNA from soil. So will it be possible at a certain day in the future that you have a cemetery and a settlement next to each other from the same period, and there will be one individual buried in the cemetery, and we will get his or her DNA from one of the houses, and we can tell that, okay, that individual actually lived there. I mean, that would be amazing. And of course, there are like cremations as well. I think we might get DNA at some point from cremations, which opens up again a lot of other possibilities in spiritual cemeteries in certain regions where we don't have inhumations, for example. So I think there are a lot of things still like ahead of us, hopefully.
Thank you, István. Krishna?
Yeah, I mean, one thing I want to mention is I think is important. I think as I tell Patrick and István many times, I don't really care about avars or anything like that. This is not particularly important to me. There's no interest. What is important is the process. In particular, thinking about how do we train the next generation of individuals? So we have people like Hannah and István here. This is a uniquely interdisciplinary subject area. If you go to NSF or NIH and do some kind of training on interdisciplinary, multidisciplinary work, there'll be like a neurologist works with an evolutionist, right? It's very different levels of interdisciplinary that the academic world is used to. And it requires a whole new framework of thinking, well, how do you train the next set of individuals, right? You're never going to have an individual that can be specialized in genomics and specialized in history and specialized in archaeology, right? That's impossible. So we need to think very carefully for those next generation. How do we make sure that the historian who has a deep level of understanding of theory of history also understands and is literate in the genetic analysis? And they can't know everything. And then you have these different groups interacting together. So that's partly what we're trying to work on a little bit with this Histogenes project and this kind of interaction. But I think that is more important than any particular question about migration. It's how do you make this a systematic process, and there's a framework of going forward, and you can put it in different areas and different time periods. And that is really going to come from the next generation of researchers and scientists.
Thank you, Krishna. Hannah?
Yeah, absolutely. I think one thing that I'm looking forward to is that so often sort of the metric that we use to talk about the growth of ancient DNA as a field has been sort of the number or the quantity of genomes that have been sequenced. And, recently I've been seeing people look at other metrics. So either the quality of them, we talked about ways to either reduce contamination or improve sort of your sequencing accuracy, or increase the coverage, which makes sort of questions that were only possible with modern data now possible with ancient data as well. And I also saw a wonderful talk by Crystal Sosi recently where she encouraged people to sort of measure and track community engagement as a metric for ancient DNA as well, which I thought was really exciting. So yeah, I'm looking forward to those other ways of sort of quantifying the field.
Well, thank you all for those comments. This has been a really interesting conversation. I wanted also to thank Tina and Tim for organizing this meeting, and I think we'll be recording a separate intro with them. We look forward to posting this and in both in an audio format and in print. And information about that will be made available in the podcast description. So take care, everyone, and carry on the good work.